This is CheloniaCast episode 81. Today we are joined by Dr. Fred Janzen, who's a professor emeritus in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. He's also the former director at the Kellogg Biological Station and a current professor in their Department of Fisheries and Wildlife. He's the PI in the Janzen Lab of Ecological and Evolutionary Herpetology, currently at KBS, formerly at Iowa State, where he does some really cutting edge and fascinating research, kind of at the intersection of a lot of different fields, but primarily sort of field biology, experimental biology, genetics, and broader ecology. To get at a variety of sort of evolutionary and integrative questions pertaining mostly to turtles. He's an incredibly accomplished scientist who, again, has used turtles as sort of a model to get at these really life's deeper questions. He's published over 200 peer-reviewed scientific articles on a lot of these different topics. He has won numerous awards for his work, including Cornell University's College of Agriculture and Life Sciences Outstanding Achievement in Research Award and various NSF grants and recognitions. He also even won the Mark Perry Galler Prize for Best PhD dissertation at the University of Chicago, his alma mater. Really, just an incredible a testament to his work. But his impact extends even beyond the research field. He's an esteemed educator. He's mentored dozens of undergraduate and graduate students in his lab. And in 1988, he started Turtle Camp, which has gone on to inspire hundreds of pre-collegiate students, where he's passed down his passion for turtles. So it's really an honor to have Dr. Jansen on today. Thank you for joining us. This is going to be awesome.
Yeah, my pleasure, Michael. Thank you, and very kind introduction. But and it has to be, clear that it's really students that are doing all the hard work, not so much me.
Awesome. Well, I think we'd like to start off with just why turtles? And
Mm.
I'm curious for you in particular, a lot of your work has been in the vein of sort of using turtles to get at broader evolutionary questions. So did that interest evolve separately, or
Mhm.
were the turtles the vehicle for that?
Yeah, it's true that turtles were a vehicle for that. But part of the thing about turtles came about a little bit roundabout. Maybe it's of interest. When I was an undergraduate, I started my research experiences working on saguaro cactus. In fact, my very first publication was on saguaro cactus physiological ecology, and I spent a lot of time in the desert, and got fascinated, of course, in between measuring cacti. Get to see a lot of really cool things in the Sonoran Desert, a lot of neat snakes and lizards, desert tortoises, all that kind of thing. Um, and so I started thinking about maybe doing physiological ecology in graduate school, and I was particularly fascinated by horned lizards. Um, but I applied to a bunch of places. I only got into one, and the person I was going to work with on lizards didn't accept me into his lab. But I got into a different lab that worked on turtles. So there's a little bit of a roundabout way. So it's not to say that I'm not interested in turtles. Obviously, I am, but that wasn't the starting point.
That's interesting. So it's
Yeah.
sort of opportunistic, and then you
Hmm.
got hooked immediately.
Yeah. Yeah.
That's awesome. So who were I mean, just going through some of your early publications, I mean, you worked with a lot of people that were very, I mean, notable people in the field in a variety of fields. I'm curious
Hmm.
who some of your early influences were and sort of what you learned from them.
Yeah. Well, I've had a fair number. My very first publication, like I said, was on saguaro cactus, and it was really important that the person that co-authored that with me happened to be around at the time when I was trying to devise this project and make it work. His name's Joe McAuliffe, and I think Joe's just retired. He was director of science at the Desert Botanical Garden near Phoenix for many years. But Joe was really instrumental in helping me stick with it and learning that process of data analysis and publication, because I was an undergraduate at a small liberal arts school, and that's not the typical experience. At least it wasn't in the dark ages back when I was an undergrad. There's a lot more, direct involvement of science nowadays in that kind of communication process. But so Joe was really instrumental in kind of kicking it off there. But when I was doing my master's degree at Colorado State University with Gary Packard, he had a graduate student named Gary Poxtus, who was also instrumental to me in helping me realize that graduate school wasn't so much about taking classes and doing the homework and going home, but it was a lot more about what you're doing in the evening and the weekends and during the day, to sort of pursue that passion that you might have for the scientific questions and the organisms of interest. And so Gary was Poxus was a real sort of spur for me to really get productive in that way. Gary Packard, my master's advisor, was too, but in a different way. His was in a much more um, uh, experimental design, statistical analysis, rigor sort of a way. Um, and then I had some other collaborators. I would say, but Poxus is probably the most influential. But John Tucker at the Illinois Natural History Survey was real big too. He's similar to Poxus and just sort of being interested in lots and lots of different kinds of organisms and systems and questions. So, we did projects on not just turtles, but zebra mussels and excuse me, different kinds of snails and all sorts of fun stuff, just because we could. And so that was kind of fun to sort of to have other people that had a, a natural history passion like that for not just for herps, but for other things and trying to figure out what's going on. That was great.
That's awesome. It's, like,
Yeah.
definitely just being
Hmm.
able to tackle a lot of different questions, and, I mean,
Hmm.
that's cool, though, that someone, that the big mentor to you was someone that I assume wasn't much older, is a little bit
Yeah.
more ahead in school, but
Correct.
that kind of person can just be
Yeah.
that thing that kinda guides you. It's like graduate school
Yeah. Hmm.
seems fun, but also, I mean, you have a lot more free time, and you have to figure out, what do I
Yeah.
do with this time?
Right.
I need to be
Yeah.
productive.
How do you productively use it? Exactly. Yeah.
Right, right.
Yeah.
Well, yeah, I mean, I think we can jump kinda into some of the things
Sure.
that you've looked at, and it's a really fun discussion because 'cause you've kind of looked at it with respect to life history, m pretty much, I mean, a
Yeah.
large quantity of things, so there's a lot to go into, but the first thing to me that
Mm,
is really kind of interesting is that with turtle eggs, the fact
mm,
that the environment, like the hydrate condition of a nest,
mhm,
can influence characteristics of that egg. I mean, that's fascinating. Why is that?
mm. Yeah, I don't think we really have a particularly good answer. I mean, I think there's arguments about how it can be adaptive because in moist soils, those eggs can um, draw uh, water from the soil to make sure that the embryos are sufficiently hydrated and they can, undertake the proper biochemistry they need for completing development and growth before hatching. Um, but at the same time, that also means they're vulnerable. They're vulnerable to desiccation if it's particularly dry, um, and really to flooding too. Um, Whether that's through taking in too much water and sort of bursting, or simply from the water preventing oxygen, gas exchange. So it's not obvious that it's necessarily adaptive, um, in my opinion, because you also have plenty of turtles, tortoises, soft-shelled turtles, and so forth, that have rigid shell legs too. And yet, you might find on the same sandbar, you might find, map turtles, snapping turtles, and soft-shelled turtles all nesting, and yet their eggshell structure is radically different. Um, yeah,
Right.
it's really amazing to me, definitely.
Do you think that it has a large impact in the field, though? And at some of the early
Mm,
experiments, you looked at pretty different water potentials, but in the field, I
mhm.
mean, I imagine it's not as variable as maybe you'd have in a laboratory.
Yeah, I think that's true. I mean, some of the treatments that I used in some of my early work were based more, or chosen, I should say, more for the idea that I could compare my results more directly to prior research than they were necessarily to, um, you know, values that are used encountered within a nest. I think there'd been some subsequent work when as the technologies improved to measure soil water potentials, that we kind of have a better sense that the soil is a lot more saturated at nest depth than we might have originally thought, and that it's not as common to have particularly dry conditions within nests that really stress the embryos in a desiccation type sense. So yeah.
Right. Okay. Yeah. So yeah, using a
Mhm.
baseline value, comparable values is not going to necessarily reflect what's in nature, is it?
Yeah, you just have to hope that those were chosen originally to sort of be
Yeah.
representative, and I think that they were based on the technology at the time. But that's true, like with the incubation chambers and the way we can monitor temperatures in nests and other things now. I mean, the technology has not only improved dramatically, but the prices are lower too. So the things we can do at scale are so much better now than they were 30, 40 years ago. It's awesome.
Right. Right. Well, that sort of brings me just talking about the influence of environment and maybe honestly genetics on
Mm-hmm.
aspects of turtle embryos. It brings me to
Mm-hmm.
something that you've worked on extensively: the environmental sex determination, temperature dependent sex determination. And it's a curious subject. There's a lot of things about it, but the first part
Yeah.
I'm curious is we think about it as this thing where the environment's influencing in some way, and we've had
Mhm.
other people sort of talk about how is it the temperature is
Yeah.
causing the sexes to develop, but how much of it is really the environment having an impact versus
Mhm.
what level of it, I guess, is heritable in
Mhm.
terms of that ratio and how it responds to temperature.
Yeah, that is actually an incredibly crucial question, especially now as environments change rapidly and organisms that have any kind of environmental sex determination, but obviously we're talking about turtles here, so it's temperature dependent sex determination. Um, not to get anthropomorphic about it, but they have to figure out a solution. um, because the way in which somebody landscapes their yard or their property is enough to change the thermal environment that a nest experiences. And it doesn't have to necessarily be at the global stage. So, how much that thermal sensitivity of developing embryos, uh, is of sex determination is heritable relative to uh, the environmental input is crucial because the more heritable variation there is, the more likely that these species or populations are going to be able to respond to natural selection on sex ratio caused by these environmental changes. And so some of the work that we've done, or I've been involved in anyways, has historically involved constant temperatures in the laboratory. That was the early work. But now we're at the stage where we're able to address these questions in actual nests in the field to look at, the inheritance or the heritability, I should say, um, of these kinds of traits, including maternal nesting behavior. But you didn't ask that part.
Yeah, yeah, we'll get into that, I think.
Yeah.
So something that underlies this is this concept of a reaction norm,
Hmm.
which, as I understand it, sort of within a clutch is sort of the variability of the response to temperature in terms of what's going to produce in that clutch, all males or all females, or that pivotal temperature, or the transitional range of temperatures. But maybe
Hmm.
so that reaction norm, the extent to which it's under
Hmm.
selection is something that you've looked at, and
Hmm.
I'm curious, I mean, A, sort of, is that to what extent is the
Hmm.
reaction arm under selection? And B, I mean, how do you even get at
Yeah.
quantifying selection?
Hmm. Yeah. That's a great question. And that's the other side of it. So, if we're talking about microevolution or evolution and response, to environmental change, it's not just about the genetics, but it's also about the strength of selection
Yeah.
acting on those phenotypes. So, in the case that you're sort of asking about of a reaction norm, right? That's basically how a particular genotype responds to any one of a series of environmental conditions to produce a phenotype. In this case, we're talking phenotype is gonadal male, gonadal female, right? And and so if you think about a clutch as sort of one genotype and you spread eggs across a series of environments, then you get a range of different sexes or sex ratios produced at any given environmental. So that you get this reaction norm. And for turtles, it's kind of looks a lot like a logistic reaction or like a logistic form, where you have males produced at cool temperatures, females at sort of really warm temperatures, and then that sort of narrow range of both sexes being produced. And so the question is, can you ask questions or can you quantify um, both the genetic basis of a trait if it's a reaction norm, or do you have to decompose that into components like only focus on that pivotal temperature, the temperature where you get a 1-to-1 sex ratio where that reaction arm pivots? Or can you look at the width of the range of environmental conditions that produces both sexes, the transitional range of temperatures? You know, so you're talking about slope and intercept. Are there other ways, or can you get an equation for the entire thing? And so these are challenges. We focused on the pivotal temperature and the transitional range of temperature for now, um, because they're quantifiable and they're the sort of thing that we think would be subject to natural selection.
Right, right. And so, I mean, you're
Mhm.
using I'm just curious. So starting out a lot of your work, you kind of use just multivariate regression analyses, like you said,
Oh, for
logistic
selection, yeah.
regression analyses for
Hmm.
selection.
Hmm.
And this is the quantitative, that I guess the analytical methods you can use to get it quantifying selection
Hmm.
have evolved. And I'm just curious from where it sort of started
Yeah.
to over the course of your career, where
Mhm.
has the ability to analyze selection sort of where has it come to, and where is it going?
Yeah, so there's some of the work that I've done, and a lot of it was well with really nice, really good collaborators, Butch Brody and Alan Moore in the sort of mid '90s, and Hal Stern in the late '90s, to try to really kind of push forward some of these methods for quantifying and visualizing and analyzing natural selection. Um, but we built on the shoulders of giants, right? So Steve Arnold and Russ Landy, who actually were both on my PhD committee, were the ones that really kind of um, brought some of the early plant and animal breeding statistical machinery into an evolutionary framework, and then we kind of built on top of that. And there's a lot of people that have done much better or more advanced work since I was doing a lot of that in the '90s, and push it forward. So I think right now what I'm seeing is two things. First, we can do it better than we used to, but at the same time, I see a lot less interest from the scientific community, the evolutionary biologists, in actually quantifying selection on traits. It's a bit of an oxymoron or a contradiction, or I'm not sure what. We can do it better, but the interest isn't there.
Why is that? I mean, why is it that is
I think yeah, that's a great question too. I suspect it's because people got um, maybe disappointed that uh, using the sort of actual estimates of selection to gain insight into systems and their evolutionary potential wasn't giving them the answers that they wanted or the clarity of the answers that they wanted. That's what I think.
that's interesting.
Hmm.
It's almost like it seems like now that, especially in the past few decades, the technological sort of advancements, there's been a lot
Yeah.
of focus on the functional basis of different traits versus
Hmm.
maybe the cumulative sort of evolution of those traits. I don't know.
Yeah, I think you're touching on the right things. The advent of genomic and other omics level kinds of analyses are the latest. They're really important. So I don't want what I'm about to say to be misconstrued because I do this too. But they're the kind of the latest shiny object. And so you get a rush of attention into that area as folks are pursuing, you know, how to apply these new technologies to some of these older questions. Um, But I think sometimes we can throw the baby out with the bathwater and put all our attention on the new shiny things, and we're going to learn new stuff, and we've already learned a lot of cool stuff for sure. But that
Right.
doesn't mean that, that these other insights. I like to think of it this way, right? The genetics and genomic side, and then there's this sort of ecology side that involves selection. And the way we're really going to understand evolution is when we put the genetics, genomics, and the environment together to get at the evolution side of things.
Right, right. And I think that's a great transition into it's just thinking about the evolution of a turtle in particular, and
Mm,
its strategies to producing offspring. I mean, there's some different. You could have evolution of the sort of the reaction norm,
mm,
a shift in that, which that sort of it entails an underlying molecular adjustment that is. I think you assume that that's a heritable thing. If
yeah,
there's
right.
a high heritability, it's probably it's something that's genetically controlled. So there's potentially more room there for selection to act on that characteristic. Is that
Or
the
to
right?
be Yeah, to be effective. So if the trait has more heritability, in other words, if it's more faithfully transmitted from parent generation to offspring generation, then as a target for natural selection, for sure, it's much uh, more effective in allowing evolution to happen, for sure.
Right, right. But then there's also the ecological side, like you were saying,
Hmm.
and still selection is going to act on this, but female behavior can influence the evolution
Oh yeah.
uh, of sort of the survival of a clutch, which is the thing that I guess is driving that evolution to some extent. But there's the female behavior in terms of, where she's nesting. Um, you've looked at this, and we kind of touched on this a little bit. I'm curious,
Hmm.
um, how much do females know? What are they analyzing when they nest? Do they know where they're trying to nest? Is it random? I mean, what goes into that?
Hmm. Man, I would love to be inside the head of a turtle to know. Exactly what's going on. I can imagine it differs between species too. Um, but I've spent a lot of years and all the folks in my team focusing on painted turtles, um, mostly because they're common, but also because they're widespread geographically and occur in environments that are radically different, thermally. So if we have a population in northern Idaho and one in south central New Mexico, I don't have to do a lot of convincing to tell you that of you that those are very different thermal environments. And yet, you've got viable painter turtle populations in both places. And so one of our big questions was to try to address these this issue of population stability is, so where are the moms nesting? What's going on there? And but from the mechanistic side, which I think you're more talking about, that it means that they have to be able to sense something in those environments so that the New Mexico population is not overproducing females and the Idaho one's not overproducing males. So they need to be able to sense something about the thermal qualities of a particular nest location. And the best we've been able to come up with is that because painted turtles are diurnal nesters, they're nesting only during the day, at least when they initiate the nesting process. They may finish after dark, but they're starting while it's daylight. Is that they're somehow sensing the canopy um, around them, not the cloudiness of the day because that's ephemeral, but something that's more permanent in the environment. But from a neurological standpoint and so forth, boy, I would love to know. I don't know if they're doing it consciously, but I'd love to know how they're doing it.
When I also think about the fact that I mean the reaction norm of a given clutch is going to
Mhm,
be different, and so does the
mm.
female. Does she know what she's doing with respect to her clutch?
Oh my God. I love this question. I'm analyzing those data right now to try to answer that question. That is hilarious. I can't believe you touched on the key thing. Does a female, right, based on her, right, the sex ratio reaction of her
Yeah.
babies, does she, when she does her nesting behavior, also bias her nesting behavior to either counteract that reaction norm or augment that
Yeah.
reaction norm, right? Yeah, that's exactly what we're trying to figure out.
I mean, I
Yeah.
maybe this is something if you're working on it now, you don't want to talk about it, but I'm
I'll
curious. I mean,
talk about it. I don't have the answer. I
yeah,
don't
yeah.
know.
How do you even start to study that? Because I'm thinking about
Yeah.
something. I mean, if you couldn't do this, but if you transplanted a clutch from a different female into another female,
Ah.
then you could get at that. But there must be another way to get at this. I'm not.
Mhm. Yeah, well, one of the ways that we've been trying to get at it is we know where the moms nest. Let's just say well, we can focus on, I don't know, the Nebraska population or something, one of our populations, and the female comes out, she chooses to nest. We can quantify the canopy cover at the time she nested, and so we have uh, a value between 0 and 100 that talks about essentially her behavior quantified that way. We'll also know, we'll put a temperature logger in the nest. We'll also know the offspring sex ratio when they hatch at the end of the summer. So, and we'll know that for a whole bunch of nests in that population. And one of the things we can do is we can ask the question is, that offspring sex ratio, how, uh, this, this is the part we're kind of struggling with actually quantifying, but, how different is it from what we would have expected based on the canopy cover value alone, the behavior, the maternal behavior? So is it more male than it ought to have been or more female than it ought to have been? And then we can ask the question, are those, um, sorry, I'm getting technical, are those residuals uh, positively or negatively correlated with maternal nesting behavior? So females, say, have preference for nesting in shady areas that should produce more boys. Do they actually produce even more boys than we would have expected based on her canopy cover choice, or fewer? Because the embryos are kind of counteracting that, their genetics, their propensity to be male. I'm sorry, it's kind of complicated, um, it, but we're able to do it because we know what all the different nests in the population are doing. We can kind of have a population level reaction norm.
That's really so you can kind of make it, yeah, using the residual, it gives you kind of a
Yeah.
distance that a specific
Mhm.
nest falls from what you would anticipate based on some kind of prediction, a
For,
model prediction of where
right,
that's yeah,
right.
that's really interesting.
So that's the way I think we're going to be analyzing it. I was hoping to get that done last week, but we ran into a snag because one of the original way we were going to calculate it has another variable that confounds it, and so we were getting a correlation, but it was almost like a mathematical inevitability because we had anyway, one of the variables was present in both numerator and denominator, so that was frustrating. I should have known that in advance, but fortunately, my postdoc Claudia caught that and reined me in.
That's yeah, that I guess that's the fun. Once you get the models to work,
Mhm.
it goes relatively smoothly. But that's the field work. The tough part is actually getting the data. The models
Oh,
is just making it so that
yeah,
it can process it, right? But
yeah, for sure.
also in terms of the heritability versus,
Mm,
I guess, phenotypic plasticity and adjustment to
yeah.
environments, like when a female turtle
Mhm.
is nesting, how much of her decision of where to nest is inherited, is something that's
Yeah.
sort of intrinsic versus how
Mhm.
much of it is she's getting out of the water and analyzing as she goes.
Mhm. Yeah. Well, what we've been able to quantify, and this is just kind of ballpark, um, is that her sort of heritability is on the order of, say, 15, maybe 20%. So that leaves a lot of the variation in nesting behavior to other factors.
Mm,
So there might be some, like, there's my point being, there's like this relatively small fraction, say 1/6, to 1/5 of where she ends up nesting is sort of an inherited component, and then the other part is maybe a little bit more about, you know, how things are that at that moment in time.
Right.
So
Right.
there's a lot of scope for that plasticity. Is kind of what I'm saying, and that's something that'd be really nice to know. And one of the things we do have is repeated in some of our longer term populations, repeated quantification of nesting behavior, and that's how we were able to get some of the heritability information as well. So there's plenty of plasticity there.
It is interesting. I think the
Mhm,
more you kind of analyze these questions, you're tempted to ask a functional question. It's almost like,
mhm,
okay,
right.
there's 80% or so of that
Yeah.
behavior that's up for
Hmm.
grabs. Now,
Hmm.
you want to
Hmm.
get into the neurology, the
Yeah.
neurobiology of the turtle to better understand how's the turtle thinking. It becomes a functional question.
Right. Yeah.
But yeah, just like
Yeah.
getting using patterns in nature to get at that is really interesting.
Hmm.
So when it comes to, I guess, the physiological characteristics maybe
Hmm.
of a clutch of turtles, and I think we'll get to
Yeah.
some
Hmm.
other aspects of this, but how much of that is like the fitness of a hatchling broadly, the swimming speed, the size, that sort of thing.
Uh,
How much we talked about this as environmentally influenced, but how much of that is, I guess, heritable?
yeah, that doesn't seem to be a whole lot more heritable in our analyses, um, which is a little bit surprising, but or it was to me, because the only reason I was doing some of that original work in terms of offspring performance was, uh, you know, one of my best friends in grad school, Butch Brody, was doing a lot of that work, performance with garter snake neonates, racetrack trials and speed, and and so I thought, well, maybe we need to look at that in turtles too. But the bottom line is, for most turtle species, with some exceptions like soft shells, I mean, escaping predators in terms of running away and doing performance doesn't make much sense. I mean, if you're a hatchling, you're pretty much going to be popcorn no matter what. But if you're older, they more or less pull in their shell. And if you're a snapping turtle or something, you're going to be aggressive. Um, so in terms of actual like locomotive performance and that sort of thing, I think uh, it's not even so much an environmentally relevant trait anymore. Swimming probably is, but not so much the running side.
Right, and okay, there's part of it, and then, I mean, the female has some control over where she's putting a nest. It depends on the distance. We talked about this a little bit.
Mhm.
Is that something that females are analyzing distance from water? Does that really make a difference? If
Yeah, it does, and we've definitely found that younger females, who are almost by definition smaller than older females, and produce smaller offspring, tend to nest closer to the shore than do the older females, who are bigger and tend to lay bigger eggs and therefore produce bigger offspring. And so one of the things we've wondered is that the case that the bigger moms, the older moms, are nesting farther from water, because that gives their offspring, even though it's a farther trek, but those offspring are larger, so they can, manage it relative to the smaller offspring of the younger moms. But at the same time, there's a potential cost, which is a lot of the predators are, um, they follow edges, and so the younger moms nest are more prone to depredation than the older moms who are nesting farther from water. So if the eggs can hatch for the younger moms that are closer to water, then they've got a nice advantage. They just have a little, you know, quick several meters, jaunt into the water. But anyway, that's a little bit about the costs and benefits and those life history trade-offs of nesting behavior.
right, and I guess some of that is maternally, as you age, you put a little more investment into nesting and reproduction more so than maybe a younger female. Is that something that nesting closer to the water in the younger females, is that
Hmm.
something that's experientially that they learn to go further, or is it just that they're willing to put more energy into leaving the water body?
That's a great question. We don't know either, but we do think there's some level of, and this is speculation, but we do think there's some level of oh, I don't know if you call it maybe skittishness. The younger moms are their first, maybe their second time on land, and you know they might not be too comfortable leaving their sort of the comfort or the security of their aquatic home. So that's a little bit more of what we think might be happening there, as opposed to something that's strategic in a different way. So it's strategic maybe in the sense of being, worrying about, having a raccoon take them, kind of thing.
Right, right. And I guess the another element to this is the maternal transfer of epigenetic sort of
Hmm.
influences and methods. I mean, how is that influencing hatchling physiology? This is a relatively new in terms of
Yeah,
the analysis of this, but I think that you're sort
yeah,
of getting at this behavior of hatchlings, that sort of thing. What have you learned about that?
yeah. We definitely. I had postdoc Rachel Bowden about 25 years ago now, and that's when we started really getting into doing a lot of the yolk hormone work, because there's a maternal effect that's transmitted across generations, that definitely has an effect, can influence some of the offspring sex, and some other kinds of traits of the developing embryos. Um, in terms of some of the other uh, more recent and more formal kinds of analyses of, say, transmitting, chromatin modifications or um, differential DNA methylation. Some of these things that are sort of transmittable across generations, but not heritable in the traditional genetic sense. Um, we think that and some other folks have shown this, in some of the early work, that they do have an impact on things like sexual differentiation of embryos. And so we're getting, anyway, we have a lot of the resources, and we're prepared to jump into a lot of that, full fledged, and trying to hire a postdoc to help spearhead some of that.
That's awesome. We a few years ago we talked to Dr. Brad Schaefer and
Oh yeah.
some of his work looking at just
Mm,
more like the methylation profiles of turtles,
mm,
profiling the entirety of it in adults and trying to link
yeah.
that, I guess, to character states and just really interesting stuff.
Hmm. Yeah.
Yeah, it's yeah, really. I'm curious about the role of microRNAs in turtles. There's been a
Mm.
little bit done with this. I don't know if this is something that your lab is looking at or
We aren't. I've been. We haven't done anything with microRNAs. That doesn't mean they're not important, though. But you can only do so much.
right, right. Well, yeah, right, right. Is that I guess we've touched on
Hmm.
the female and her choice and where she's going to nest and her investment in the fitness of her offspring. This is sort of encompassed, I guess, in phenology, in
Mm,
timing of nesting,
yeah.
and
Yeah. The
that's
maternal.
I guess that's something that selection may be acting on to a various extent. And it seems like in some of there was a big holistic analysis you did on this with a lot of other
Hmm.
people, and it seemed like the slider turtles were had the biggest it
Yeah.
shifted a lot based on climate,
Hmm.
but other turtles not so much. I mean, what is it about the sliders and maybe
Hmm.
just about maternal investment and fitness of hatchlings and how they can, alter this phenology? Why do they have that sort of plastic response?
Yeah, I think for the sliders, that was some of the thing that really, helped me, uh, or prompted me to try to corral a bunch of other collaborators to compile our data to try to do this larger scale analysis of phenology of lots of different North American species and different populations of the same species. Because some of the work that was John Tucker's primary population in Illinois with those sliders, and we kind of came to the initial conclusion before we got all the rest of the collaborators together, was that those sliders are really right at the northern edge of their natural range within North America. And we thought, okay, like many species, not just turtles, the range edges are probably largely determined, unless it's by something hard like a mountain range or a river or something. They're probably determined by environmental factors like temperature or something along those lines. And if you're right at the northern range edge, um, you know, if you're going to produce viable offspring, there's really going to be strong selection for those animals to be responsive to the local environmental conditions. So that was our hypothesis and why they advanced so much more than populations more in the center of the range of any given species.
I was looking at it interested just in terms of their invasibility, but I guess a lot of that is just
Mm,
due to the fecundity but if there's some inherent,
yeah.
if they have a more
Ah,
if the nesting characteristics of a slider, a female slider, and that sort of thing are more, I guess, heritable or genetically linked in
ah,
some way, maybe they could be a little more plastic in that sense when in a
mhm.
new environment. I don't know if that's logical.
Yeah, I know what you're saying. I really think that the answer for sliders and their global invasibility, though, is the fact that they're just so generalist.
Mhm.
Like their habitat preferences seem really broad, and their food preferences seem really broad. You know, I think I think that has a lot more to do with it, personally.
Right. And so you've also, I mean, this was something I worked on a project with southwestern pond turtles and sliders back
Mm,
in
mhm.
Southern California.
Oh, yeah.
And as part of this, we did some in collaboration with John Mayer's, the Mayer's Lab. We
Oh,
ran
yeah.
these bays and these hierarchical models. Look at the just the number of turtles in this creek system and the
Okay.
sliders and pond turtles. But as part of this, I went back and did like a vortex population viability
Yeah.
analysis and I used some of but I had to find parameters to put in for the fecundity of the turtle. So I
Ah.
used some of the data from one of your analyses with John Tucker,
Yeah.
and so but you seemed to you that was really the only analysis I could find that had good
Hmm.
data that could give me an idea for the fecundity of the sliders. I mean, so what
Wow.
is that number that you have offhand in terms of how many eggs do you think that they're laying per season?
Yeah, in an average year, they're dropping a couple of clutches, for sure, and more frequently than not now, they're laying 3 clutches a year, which is really insane. We're talking like 35 or more eggs per year, um, distributed in, like 2-week kind of internesting intervals, so it's kind of astonishing. Yeah. They're really fecund in that way, and the fact that they can spread it out as opposed to a snapping turtle that might, drop 35 or 40 eggs, but one time in 1 year in one spot, right?
Right, right. Yeah, it
All
was really
all the
interesting.
eggs in one basket kind of thing for them.
Yes, and the pond turtles, I mean, it was a yeah, right, they usually catch once, it seems like, and they'll lay maybe 7 eggs in a clutch. It's
Right?
just you're gonna get
Can't
swamped.
compete. Yeah.
No,
Yeah.
right.
Yeah. I bet your vortex analysis was pretty sobering.
Yeah, no, it definitely it was not great to see
Mm.
the problem with it, I think, with a lot of this, the population viability analysis is it's so broad brushstroke. I
Oh,
didn't have site specific data, and I had to pull this data from studies like yours, and I don't know
yeah.
how the
Yeah.
parameters of a slider population in central Illinois compare to those in a introduced urban stream in
Yeah.
Southern California. So
Fair.
it's assumption based, but yeah, the sliders shoot off to infinity very rapidly, and the pond turtles crash. The pond
Yeah.
turtles crash in about 40 years.
Wow.
So,
Wow.
that it's yeah,
Yeah.
it's
Mhm.
not a great thing to see.
Right.
Um,
And for all you know, they'd crash sooner because the vortex analyses don't even include the genetic side. So they might succumb to inbreeding and so forth even before 40.
that's a great point. That was not something, it's
Hmm.
an option, but I just there's no data for that. But that would be
Yeah.
it'd be a good
Yeah.
thing to analyze, like a different kind of
Hmm.
uh a level to that analysis, get some genetic data, try to, figure
Hmm.
out some of the parameters there. That's a really good thing. Yeah, talking about the genetic influence, I guess
Hmm.
the influence of a male turtle on a female is interesting, and it's species
Hmm.
of sperm storage, that kind of thing. You had a lot of complexity to it, but I'm curious how much we've talked about this on the podcast in the past, like
Hmm.
maternal choice, female choice in terms
Hmm.
of who she's mating with,
Yeah.
or how she's even selecting sperm to some extent. If she's
Mhm.
drawing from stored sperm or what, how that works in a female. How much does it matter?
Yeah, well, it can matter tremendously, and I just wanted to say that I had um, really awesome collaborators from University of Georgia back in the day, John Avis and Devin Pierce was a graduate student with John, and and when they wanted to collaborate to try to address some of these questions, it was just such a great collaboration. Um, because the stuff that Devin was finding out about the levels of multiple paternity and how that relates to some of these other kind of traits and so forth was just really super eye-opening. The idea that these females could, for a minimum of at least 3 years, store viable sperm from the same male and not necessarily have to remate was just jaw-dropping. I wouldn't have had no idea. And that that 30% on average of the nests in any given year are multiply sired. So these females, but I bring this up because to me it was astonishing at the level that females could be independent of males. That's not completely, but substantially, and then you know potentially do what you were saying is differentially allocate sperm from different males to different eggs and different clutches. So so the neat thing to me was that it opened up that as a possibility in a way that I hadn't appreciated before. But now, is there evidence that they do and that there's a fitness benefit to multiply sired clutches or things like that, improved hatching success or some other kind of fitness benefit? We haven't seen that.
Right. Yeah, it's the I guess the sexy sun hypothesis. This is something
Hmm.
that's come up before. It, yeah, how is a female choosing who to mate with? Is she making
Right.
that decision? How is she choosing even the
Hmm.
sperm? Internally,
Yeah. Right.
is that yeah? That's
Hmm.
another tough one to get at, but it's an interesting
Yeah.
thing to ask anyway. It's like that, like you said, the sperm storage. I could see that as
Right.
being a method to sort of, okay, this most recent is not good, so I'm gonna pull from
Yeah.
this
Hmm.
reserve almost.
Right.
Is it
Right.
okay,
Yeah.
yeah,
Or
and
I think it's not a bad way to think about how they can colonize new environments too. If you have a female that goes across the land and ends up in a body of water where there isn't another individual of her species, in particular no male, well, she could reproduce for multiple years without having to have anybody else and produce viable offspring.
right, right. Well, that makes a lot of sense in terms of right. If you that would be interesting to look at is the
Hmm.
propensity for a female, because in the snapping turtles, I mean, they have these nesting migrations, like they move long
Yeah.
distances. I wonder if there are any of those turtles that keep dispersing or they stay in those new sites. If there was a
Hmm.
correlation between that and who has stored sperm, you could see if they're
Ah,
feeding different
yeah.
sites or and moving longer distance.
Yeah. I see what you're saying. Sure. Yeah.
But
It
then
opens
I
up
guess
lots of fun questions like that. Yeah.
yeah, that I don't know how much that has merit though, because you assume in a population where you've got a lot of male snapping turtles, there might not be a need to store. But then at the same rate, I mean, the
Mm,
female doesn't know that, so it may be
yeah.
useful to have that reserve, kind of like in a migratory sense. Well, that's yeah, it's interesting.
Yeah.
I want to return back to TSD
Okay.
because I think we touched on that
Mhm.
and the selective basis of it, but
Mm,
I mean, it seems like why is it? We talked to Dr. Valenzuela on a previous podcast just about
mm,
the evolution of it, but I'm curious.
mm.
It seems like it would not be something beneficial for the most part in terms of, but what is the I guess the evolutionary basis of TSD and also the
Mhm.
utility of it?
Yeah, yeah, it's a bit of a tricky question, right? It's an adaptive significance question, and a lot of people have spent a lot of time exploring possibilities. And you know, other than in particular, I'm thinking um, Dan Warner's PhD work with Rick Shine. It was published on Jackie dragons in 2008 in Nature. able to show essentially that the incubation temperatures that produce male offspring, males from those temperatures sired more offspring of the next generation than did males that were produced at primarily female producing temperatures. And so there's something intrinsic about how temperature influence maleness and
Mhm.
femaleness to improve the fitness of each sex in the next generation. Now, physiologically and so forth, how did that work? I don't know the answer. No one knows that answer. But the fact is, they found this really, really important relationship between uh, incubation temperature and sex and sex specific fitness. That has not been so easy to come by in turtles. Um, we've done a lot of different experiments to try to look at that kind of question. Haven't found anything that's jumped out. We did have one paper where it seems that how extreme the difference is in differential age of maturity for the two sexes does seem to have some relationship there to uh, temperature dependent sex determination. Um, but it's not obvious. Yeah, it's not an obvious answer. So sorry, it's such a fascinating question because no one has a good answer to it either. It's frustrating too.
Right. The survival to maturity, that
Hmm.
hypothesis is really
Yeah,
it's
yeah, that's
interesting.
the one I was referring to. Yeah.
Yeah, I mean, it's like a correlational sort of thing, but what is the
Hmm.
underlying I mean, what's the logic behind why it would matter when you've got this bigger magnitude in age at maturity between the sexes that you would
Hmm.
have temperature dependent sex determination?
Because you have to produce individuals that are more likely to survive if they have to live long or longer till they can actually reproduce.
Right,
uh,
right.
and so that's sort of uh, whatever that temperature dependent benefit is. um, but underlying that, what is it? I don't know the answer to that again. um, but
Yeah,
but it's a mathematical, like it's kind of a mathematical consequence. uh,
right, okay, so it's sort of a it occurs because of something kind of in the that's interesting. Yeah, because I mean, there's the logic behind it is like because you've looked at how different temperatures influence the physiology of hatchlings, and you might expect, okay, if a female needs to get bigger, then it would be beneficial for her to incubate at a temperature that allows her to be a little bit faster or has some differential fitness benefit
hmm.
for her
Yeah,
so she can get larger faster, but I don't
yeah.
know if that's I don't think that that was 100% that the underlying basis of that model.
Yeah, and that's not really, yeah, that's the thing, a lot of folks have done those kind of experiments looking
Yeah.
at it from, early life perspective because, it's hard to rear animals in large numbers for a long time to be able to kind of quantitatively address that question. And ideally you'd raise them to maturity and so on, like Dan did with the Jackie dragons. He had the good fortune that they mature within a year. So we're not going to get that with turtles.
Yeah,
Um,
that's the challenging part. There was
yeah.
another it was, it had to do it linked to I think it was Reinald's hypothesis. It links,
Oh yeah. Mhm.
like the natal homing with a
Mhm.
female, a female knows where she was produced to some extent, and then that
Mhm.
reciprocates. I mean, what's the merit of that?
Yeah, I mean, it was a really interesting idea. Nicole Valenzuela and I tested that hypothesis and didn't find support for it. And really, I think the big challenge in that one is, um, you know, if a female hatches from a nest from a particular beach and she survives to mature and she comes back to that natal beach, you know, that's great. But then how do you explain the production of male offspring, right? It's sort of not obvious
Yeah.
because they they don't imprint because they aren't coming back to nest. So I don't really know.
Yeah, that was the thing that stood out to me is
Mhm.
how do you where are the males coming from? It seems like
Right.
yeah.
Exactly. Are they just accidents? And so you expect very female biased populations with a handful of males here and there, enough to, sire nest and so forth, or sorry, sire females,
Right.
and but we don't see those kind of biased sex ratios.
Right. Right. Well, and this brings up, I guess, a good with the changing climate, and that
Hmm.
just the environment that's changing. What is the thinking about TSD in evolutionary context? It's still, like you said, we don't know what the functional, the significance, what the benefit of it is,
Yeah,
why it would have evolved, but it exists, and it's just curious
yeah,
to think in this changing climate. It seems like this could be a maladaptive characteristic. Is this something we talked about the magnitude of selection
yeah,
on the trait itself
yeah.
is not very it's not really there. There's not a lot. The magnitude is small. It's small magnitude.
The heritability part.
The heritability part of it,
Yeah,
sure, yeah,
right.
yeah.
Hmm.
But when it comes to climate change, I mean, how is the climate going to
Hmm.
impact are turtles, I guess, going to be able to evolve rapidly enough to counteract the impacts of climate change? And what does that evolution look like?
Yeah,
That's what I'm getting at.
yeah, yeah, and because when you do look at it from the perspective of heritability of traits that might influence offspring sex ratio, those heritabilities, like we've discussed, are low. So that doesn't provide a lot of scope for sort of traditional cross generational evolutionary change. Um, and so we've started looking at it from sort of two perspectives. So one is sort of a deep time macroevolutionary one. Um, and I'm just going to sort of summarize it as, historically, how did turtles with TSD withstand, major environmental upheavals? Because they existed over the last 200 million years, they did,
Yeah, yeah,
right? So what's happened? And, a lot of the possible answers come around to the fact that they could migrate um, in an environment, in a world uh, that didn't have interstate highways and locks and dams and all the kind of things that we have nowadays that kind of prevent, other than for sea turtles, mind you, that really kind of prevent their capacity to move. And the rate at which the environment's changing currently is probably faster than it has happened in most of those other times as well. So that combination is not great. So the way that we think that they're kind of solving the problem now, um, at least in the short term, buying time for evolution, if you will, is with plasticity in their nesting behavior. So we've done two different things. One is a sort of an observational study, and the other is an experiment. So we brought moms in from populations. This is a painter turtle study. We brought moms in from all over the geographic range to one common location, and we provided them with an opportunity to choose where to nest. And we were asking the question, do they choose to nest in areas that are like those in their home population, or do they choose to nest in sort of shady area or in relatively shady areas that are characteristic of their current location? This was in Iowa. So did they nest like the local painted turtles nest? And unbelievably, that's exactly what they did. So the turtles from New Mexico, the ones from Washington, the ones from all over the place, they nested just like the ones in Iowa did. It was
right,
it was unbelievable. So they're in that sense they were almost completely plastic. So they did what was necessary to, I don't know, I'm putting it in air quotes, right, to be adapted like the local population is. So that's one kind of way. If your moms across their geographic range can nest or choose nests that are going to have a thermal environment when sex is being determined that's just like the thermal environment in a nest in any other location throughout the range, no matter what the macro environment is, right? What the local climate is, how hot it is, and so forth. But if the moms can manipulate by this plasticity behavior, then your offspring aren't going to be subject to this environmentally induced sex ratio selection. They're shielded.
right, right. Yeah, it's just such a fascinating question to try to wrap your head around is why
Mhm.
the why behind it is like how do you Yeah, like, is it something where the turtles are buying time? So is
Yeah.
dispersal in turtles? I mean, you think about this with translocation of tortoises and different turtles.
Mm.
Like
Hmm.
they'll if you
Hmm.
move them to somewhere they don't want to be, they move long distances, and then
For sure.
sometimes you'll track an individual that will move tens of kilometers away from an
Right.
area for whatever
Yeah.
reason. It's like they've
Hmm.
got this
Hmm.
intrinsic propensity to move in certain
Yeah.
individuals.
Yep.
But I mean, a lot of animals have that. But how does that relate to are they constantly I think what you're sort of getting at, the turtles are
Hmm.
constantly slowly kind of adapting behavior to mirror changes in temperature. Is that the
Yeah, I suspect that that's true, but but I can also say that we've been studying these things for almost 40 years now, like some populations over that long term, and it's not obvious that the overall uh, way that they're choosing the environment has changed. So it's not like they're choosing shadier locations now, 40 years later than they were, in the 80s.
right,
So, but on the other hand, I mean, how much has our global environment really changed in the last 40 years? A degree and a half? That's not a lot. And so maybe we don't expect to be able to detect it yet. Maybe we're asking too much at this point. And so, you know, it, so so what we did with the painted turtles thing was to substitute essentially space for time. We don't have time. We have 40 years, but that's not long enough for us to detect a change, say, within a location. But if we can ask, how populations in these radically different climates now, how they solve the problem, that might give us insight into how populations in a given place will solve it through time.
right. Yeah, it's just there's so many different aspects to the evolution of TSD. It's fascinating to think about.
Yeah,
The long term, it's tough as a
yeah,
human, someone that's here for maybe 100 years, thinking in
right,
these long term
yeah,
scales. Is
hmm,
there a broader pattern that we just don't have the temporal understanding to
yeah,
grasp?
right,
Is really an interesting question. I'm curious
yeah,
too. I mean,
yeah,
habitat loss
hmm,
is
ah,
impacting
yeah,
turtles and obviously
everything,
just through direct mortality, but
yeah,
in other more subtle ways. And you've looked
hmm,
at this, I think, with snapping turtles, is like the difference in temperature in an urban nest can be
yeah,
really quite extreme. I
yep,
mean, what
yeah.
does that how is that going to impact the response of TSD?
Yeah, and the snapping turtles, I think, are particularly an interesting case because at least the ones that I'm familiar with are nesting mostly, initiating nests uh, uh, after dark or in very dim light situations. So I don't think they're assessing the thermal environment in the same way that so many other turtles do. Um, and so they're particularly susceptible to that sort of a thing. They're choosing nests on more, I guess, I'm not sure. We think that they're choosing them. They tend to choose sandier, more open locations, but not any sort of taking into account, canopy or whether they're next to a building or any of that kind of stuff. And yeah, I think that kind of ecological trap is going to be really disastrous for those species, for
Right.
sure.
Right, and
Yeah,
that's still the level of are they gonna start nesting in those sites less if it becomes an issue, just the phenotypic, the plastic response, like,
hmm,
that's maybe we
right.
won't see that on the short-term timescales.
Yeah, I don't think for snapping turtles you will. And then you're going to need probably a couple centuries to answer that question.
Right, right. Well, that's I think we can sort of transition. Okay, now we've talked a lot about the making of the nest and the female's role in that. But when you have the hatchling turtles and the turtles actually in the nest, and it comes time that they've
Mm,
developed to the point where they need to leave, there's a lot that goes into that. And the first thing that stands out to me is this synchronicity in
mm.
hatching and pipping. And I'm curious. I mean, what's the benefit to that?
Yeah, so I want to give a shout out to Ricky Spencer, who was a postdoc in my lab and kind of got me turned on to that possibility because I hadn't really ever thought about it too much, other than I was familiar with it from the sea turtle literature, but I hadn't really thought about it from the sort of, I guess, freshwater turtle perspective. Um, and so Ricky helped us design these really neat incubation experiments to look at, sort of mechanisms of promoting synchrony and hatching. But to get more to your question about benefit, I think from what we were trying to assess was whether there's a group benefit. So if you emerge from a nest as, a dozen or 40, depending on how many, siblings you've got, um, and you're emerging, at the same time, the likelihood that a predator is going to eat all of you is a lot lower than on a per capita basis than it is if you emerge just a few at a time over a span of multiple days. And then you're just giving that raccoon or that crow or that heron, you know, a meal every day, and then you're all gone. But if you can sort of, swamp the predator by coming out all at once, your per capita odds of getting taken are lower. So that's kind of what we think, and we have some evidence for that.
It certainly seems like it in the sea turtles. I've seen
Mhm.
the boil up. I had the fortune of seeing
Oh,
that.
cool.
Yeah, and it's just it's a mad dash. I mean, it
Yeah,
was yeah, it seems like that seems like it would be I mean, not always the biology isn't always that what it appears, but
sure.
yeah, that seems like a logical place
Right.
for that. Something that I've been thinking about this a lot recently is this bigger is better hypothesis about the
Hmm.
hatchling turtle, like
Hmm.
writing the snapping turtle overview. eh, That comes up a lot, and it's kind of been a hot topic. A lot of different people have different
Hmm.
views on this, and it seems like
Hmm.
even throughout your work,
Hmm.
the results you've seen with this have changed. So,
They vary. Yeah.
yeah, do you
Mhm.
is bigger better in a hatchling turtle, and does that depend on species?
Yes, and yes. It does. Yeah. I think it depends on even within a species. I think it depends on context too. And by that, I'm mostly referring to environmental context, the local microenvironment, and the weather conditions that transpired during the course of migration. I think those matter quite a bit. But yeah, we've done some, I think, really neat large scale field manipulations using the slider system in particular to be able to disentangle causes. We don't seem to see much in the way of things like desiccation or stuff like that causing differential mortality based on size. Um, but in brief, it seems to be about the predators. Um,
Right,
and the size thing comes in not so much as being big enough so that a predator has difficulty handling you, as much as the bigger you are, the faster you are, and therefore the less time you spend exposed on land to that predator. So that's kind of how the big comes in as a proxy for being faster, really, for
right,
performance.
right. And yeah, I mean, I think of it this like, what are the constraints? I mean, I guess an egg, yeah, the optimal egg size is right. I'm thinking of
Hmm.
just what are the it seems like if that was the case, you would want a huge hatchling turtle, but there's going to be constraints on the size of female, the egg a female can lay,
True.
and that's you've looked at this the optimal egg size
Hmm.
theory. I guess if a
Hmm.
there's some alternatives to this, but it seems logical. A female's going to produce. There's sort of a trade-off between clutch size and egg size.
Right.
But how does that manifest
Hmm.
in systems that you've worked in?
Yeah, it's one of those things where from the maternal perspective, producing more offspring is better. I mean, mathematically, that's just the truth. From the offspring's perspective, being tiny is not so great. Because, uh, and so, um, there's from the offspring perspective, they want to be larger so that each individual would like to survive. But again, from the mom's size, then that means she's producing fewer offspring. So
Right,
that's the maternal offspring conflict over size. And as a consequence of that, right? One generation pushing to be larger, the other generation pushing that one to be smaller. There's going to be somewhere in there, I don't know, I guess call it a happy medium for sake of conversation. And that's mathematically is you can solve for what that optimal egg size ought to be. And then you can ask, what is the actual egg size? Does it match that? And we found that it turns out that the optimal egg size tends to favor what the moms like relative to what the kids want.
Right, so they're producing a little bit more than you would expect at a smaller egg size. It's
Yeah,
kinda yeah, that's it
yeah. It's kind of anyway. It was pretty fascinating. It was a fun project.
yeah. Well, and speaking of the field aspect of this, I mean, a lot of this
Hmm.
you worked with drift fences and you're
Hmm.
monitoring these fences, for a long period of time. What was it like being out in the field doing that? I mean, that's rigorous work.
Yeah, yeah. It's a challenge, and to be able to really install these things took so many person hours, so many people helping out. It's just really been awesome to do it. We've got another one we're putting in a few weeks to run yet another pair of experiments. Releases, doing these release, we call them release experiments. Um, uh, and yeah, they're a lot of work. And I will say the very first ones that I put in when I was just more or less by myself doing it, I was pretty nervous about whether I'd catch anything. I have to tell you, I thought, my God, they can go anywhere they want, um, and and yet they didn't. So it was yeah, shockingly successful experiment
That brings up
for
something.
field work. Yeah.
Can the turtles see? I mean, I've seen
Mm.
drift fences before, but maybe the system you use is different. Can they see the
Mhm.
water from the drift fence?
I mean, not when they're really close to the drift fence, but otherwise they'd be able to for sure
Okay,
because it's
yeah,
close enough. Yeah,
yeah,
to the they ought to be
yeah,
able to see it until they get to the within the last few feet maybe.
yeah. Because the water finding ability in turtles is interesting,
Hmm.
and that could be
Yeah.
it'd be
Did you talk to
interesting.
Justin? Did you talk to Justin Congdon yet?
Yeah, yeah, we spoke
Oh,
to
good.
him,
Yeah.
at the TSA meeting. We got to do
Oh,
a
okay.
live one, so that was
Oh,
great, but
cool. Yeah,
yeah.
because he's done some of that work with the
Yeah,
water
and
finding.
we spoke to him about that, but I would just say I mean, that could be an interesting thing to look at is a different
Mm.
size, like, try to obscure their view and see what the proportion of
Ah.
hatchlings that make it versus not. I
Wait, are you gonna put face masks on them?
well, yeah, I don't know, the
Yeah.
logistics of it may just make a
Mhm.
really tall drift fence is kinda what I'm
Yeah.
thinking, but yeah, you could, I mean, honestly,
Yeah.
that that's it's sort of a crazy idea, but 3D printing and things now, I mean, you could probably
Hmm.
obscure a turtle's view, but,
Yeah. The
yeah,
reason
that's
I well, the reason I ask is because you're talking about a higher drift fence, which is one way. But what if they can sense the reflection of the water into the lower atmosphere, right? So you kind of need to obscure their total vision because, like,
right.
anyway, just wondering if they could do it indirectly, sense water that way.
That's a good point, and I mean, you could do it this may be something that you wouldn't have to do with a hatchling turtle, you could do it with an
Mhm.
adult, I mean, that'd be easier to put a mask on an adult and then just release it, but then
Mhm.
the hatchlings are sort of a blank slate,
Ah,
so you've
true.
gotta deal with that,
Yeah,
yeah,
good point. Yep,
but yeah,
exactly.
getting at that, it's a like, because what Dr. Condon's done
Mhm.
is, to my knowledge, mostly the sort of, the maintenance of a directional heading using the
Yeah,
sun
the
totals.
orientation. Yeah.
But how
Mhm.
do they know this is the thing to me is that's
Mhm.
fascinating is how do they know where the water is? Like, they know how to
Yeah.
maintain it, but they need to know
Ah,
where to go, and the
right.
sun doesn't necessarily tell you all the
Mhm.
information about that. So I think it
Mhm.
maybe that
Yeah.
could get at something. That's some
Mhm.
experimental design. But, yeah, I
Yeah.
mean, regardless, it's fun to talk, like, there's the results, but then being in the field is something different and extremely fun. So,
Yeah.
uh,
Yeah.
something related to the hatching, I guess is like just passively, have you observed any sort of communication or indication of
Mm.
socialization in turtles broadly and in hatching?
I haven't, but I don't have a setup that's um, probably really ideal for being able to detect that. Our incubators, for example, make noise. Our environmental chambers and so forth, um, and if they're making noises that are relatively quiet or maybe high frequency, I don't know how we would detect it. And I'm probably the worst person practically among many to do it because I have a hearing disability that I've never even been able to hear sounds like that personally.
Oh, okay.
Yeah,
Yeah.
I was
That's
kind of born without, most of my hearing. So
dang. Okay. Well, yeah, that's a challenging
yeah,
thing to
so there might be
analyze.
that area of turtle stuff I probably will never seek to try to understand unless somebody in my lab wants to do it.
Yeah. Yeah. Well, yeah. I mean, that's interesting. Yeah, and I guess yeah, in hatchlings, there's a lot to tackle here, but
Mm,
in the painted turtles, the super cooling. I don't think we've ever really focused on this, but you've looked at
no.
this and um, it's just fascinating. Up to
Hmm.
I think about -9
Yeah,
Celsius is it,
yeah,
and they can. What is the thing
yeah.
that allows them to do this?
Yeah, that's not something that we've examined really in my lab, but my understanding from Gary Packard work and others, and John Costanzo and so forth, is that they have these large scalation on their forelimbs, and so when as the hatchings are pulled into their shell, they're kind of uh, it would be like being covered with a lot of fingernail material. It doesn't transmit the 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 ice crystals into the body so that they're not turning into a turtle cube. And so they're kind of the shell and these, I can't remember what they're called, dermal something, uh, prevent the transmission of ice into the body. So I don't know if it's anything physiological as much as it's physical.
That's fat. Okay, that's really
I
interesting.
think, I think, but it needs a lot more work. That whole area for sure.
Yeah, it seems like there's a lot of the molecular side of it. I mean, it hasn't been as analyzed, just as fully
Agreed. Yeah.
analyzed as it.
Yeah.
Yeah,
Ken Story retired, John Costanzo and Rick Lee retired. I don't know if there's anybody looking at
looking at that.
that question of, cold tolerance, whether it's supercooling or freeze tolerance in neonatal turtles at this point. From the molecular standpoint, I think it would be awesome. It would be a beautiful, wide open area now that we have genomic tools too.
Yes, yeah, that actually leads into something interesting.
Hmm.
With so you worked on the painted turtle genome. That was a really
Hmm.
a
Yeah.
comprehensive project, and that was just a fascinating. uh,
Yeah. That was
Yeah, I've
great.
been through that analysis. So just looking at because you not
Hmm.
only sequenced it with like 93% coverage, I think it was something
Okay.
really
I
pretty
don't remember.
high.
It was good. Yeah.
Yeah, but also, I mean, the thing that was really stood out about it is it wasn't just a kind of a summary of the genome, but it was you really went in to analyze a lot of the specific genes and pathways that could relate to other organisms, and like really highlighting why the genome was important to
Hmm.
sequence. And yeah, so when it comes to just freeze tolerance, I guess other thing broadly, what do you think about the
Yeah.
utility of using turtles as sort of biological reserves for other species to learn about other species?
Oh, I think they're great from a comparative sense. I think from the longest, and we haven't touched on this, but we're also doing that in the context of aging and lifespan, since aging and lifespan in many turtle species is tremendously similar to that in humans. So I think it's true. Historically, the phylogenetics of amniote species wasn't really well worked out and well understood. And I think we know a lot more that turtles are not so far distantly related from other taxa as we used to think they were. Right? They kind of share uh, a grouping with crocodilians and birds, for example. I mean, it's kind of amazing to think that turtles are more closely related to birds than they are to, say, lizards. Right?
Yeah,
It's just
yeah.
not the way historically people thought about things. So I only bring it up because I think from a comparative standpoint, they're actually well positioned phylogenetically to provide that insight. Yeah.
Right, right. There were some, I mean, just that's something that for me is I think that there's been a decent amount of analysis on the ecological role of turtles, and a lot of times we use that
Hmm.
as justification. But I feel like it seems like there's a lot that could be sort of synthesized to highlight the importance of turtles broadly across science to inform human biology, medicine.
Hmm.
That's something where yeah, it doesn't get as much attention, but even just talking to people like Dr. Schaefer, Dr. Georgia's,
Hmm.
Dr. Valenzuela, all of these different people there's all there's so much here. There's a wealth of biological information that seems like we can tap into. Is good. When it comes to the aging, that's a great
Hmm.
question.
Yeah,
And you've worked on for at least the painted turtles for, I
yeah,
mean,
right.
study sites for decades. So you've got
Hmm.
really long term data. But are turtles? It seems like this is sort of conflicting. Are they actually senescing?
Hmm.
Is there an impact of age on the sort of, I guess, life history characteristics? You know, is there an impact or not?
Yeah, in the painted turtle populations where we've been following individuals that are, sort of through their lifetime, individuals of known age, I think the thing that really stood out about that work to me was on the female side of things, where that we hadn't detected simply from the fieldwork alone. It only was once we started to look at the actual data, and it was that these females are going along year after year, decade after decade. They're coming out of the water maybe annually, laying a nest, maybe two, producing offspring, so on and so forth. They come back, maybe they've grown a millimeter, and it's like, shell size, and it's like year after year and so forth. And then at some point, yeah, you don't see her anymore, but there's a couple hundred other females in this population. And so you don't pick out the individuals per se. But when you look at the data, what we found was that in the year, right before we never saw her again, her eggs, I can't remember what it was. I think it was the hatching success of the nest was really low, and she hardly laid as many eggs, or they were a smaller size. I'm actually mixing that up. I'm not sure. But the point is, there was this sign that she'd been fine for all these years. And then all of a sudden, right before she disappeared forever, her reproductive output was compromised. And so there was this, it's almost like a cliff, basically. She's fine. Maybe it's not even a cliff in some sense that, she's got an upward trajectory, producing bigger eggs, maybe some more eggs. And then all of a sudden, boom, the end. No idea why. So I'm fascinated I mean, I don't know how you know, other than we only have this one little signal toward the very end.
Right. Yeah, that was I think I know what you're it's one of the Warner papers, I think. It
Yeah,
shows it's like
yeah,
a parabolic curve, and then right at the tail end it slopes down a little bit. And then that was
yeah, yeah. It's a pretty sharp drop.
yeah. That's well, it's really fascinating. It's like
Hmm.
they think about the telomeres and things related to
Hmm.
aging. Like, is there something there that it just
Hmm.
spontaneously shrinks or
Right.
cuts off? I don't know. Is it
Yeah. I would love to know. And we've done things like we take blood samples repeatedly from these moms, year after year or so. We probably have the material in the ultra cold freezer to do it. To answer
right?
that question. Um, but you just need somebody to do it.
No, that's awesome. Something else too. You mentioned the phylogeny of turtles. This
Hmm.
is your interests have spanned so many different things. It's tough to pinpoint where to
Yeah.
go. But yeah, so you worked on the 2005 phylogeny, one of the first
Mm.
really big global phylogeny of.
Hmm.
Well, I guess there was. I
Yeah.
think this.
90s.
Yeah, there were some in the '90s, Dr. Schaefer's,
Yeah.
but,
Yeah. Hmm.
yeah, this was one of with the nuclear, I
Hmm.
think, what, Rag 35 is a nuclear
Uh, Rag one.
or Rag okay, Rag 1, yeah,
Yeah.
so with a nuclear gene,
Hmm.
you looked at the phylogeny of turtles, what were the things that stood out to you the most about that? And I guess knowing what you know now.
Hmm. Yeah. So I did my PhD at the University of Chicago, and I realized that after my training there, that if I really wanted to uh, be a more well-rounded, comprehensive sort of evolutionary biologist, I needed to get up to speed on phylogenetics and molecular techniques at the time. You know, we didn't really have, thermal cyclers or PCR at that time, so that gives you a feel for where things stood. Um, and so I actually did my postdoc with Brad Schaefer when Brad was at UC Davis, and that was the point of it, was to get myself up to speed on phylogenetics and all that kind of thing.
That's
So
awesome.
that, But at the time, the way it was mostly focused was on there was morphology, but also a lot of mitochondrial DNA kind of thing. And so it wasn't, again, for quite a while, that we had good nuclear markers. And so a colleague at Iowa State at the time, Gavin Naylor, suggested, because he was working on sharks actually, had suggested this RAG1 gene as a good single copy nuclear gene that might evolve enough at a rate where we could gain insight into some of the controversial, branching points within the extant turtle phylogeny. So I had a master student, Jim Krenz, and Jim took it and ran with it. And so that's kind of the genesis of that particular project. And to me, I think what was so great was that, nowadays you'd probably never be able to publish that because it's a single gene kind of thing. But and yet that tree is robust. I don't think there's been much in the way of any changes to that core
Yeah.
family level structure.
Yeah.
But
That
so to me, that was fascinating. That like, wow, look at this. This is solid,
Yeah, yeah, right, even the placement of the Platysternidae and all of that,
right?
it's
Yeah,
yeah, I
yeah.
was looking at that, I was thinking, yeah, yeah,
Mhm.
this is very similar to the Thompson
Thompson,
2021. That it's very similar, and based on a
right?
single gene, the Thompson was I mean, that I think that was rad-seq is
Hmm. Yeah,
hundreds of thousands of SNPs, so
right.
yeah, very, very high
Hmm.
coverage
Yeah,
and yeah.
yeah, and they have more species in it too, so they're trying to do more work with their data for sure. Whereas we were at the family level, but yeah, but otherwise that core structure, totally, totally the
Yeah.
same.
Well, that's awesome,
Yeah.
yeah, I mean, it's just all of these different related to it's just fascinating to think about, yeah, what can the turtles teach us about concepts
Hmm.
like aging and just our general biology, and, I mean, cancer research, that's even something, is it potentially an avenue
Hmm.
there. I guess the to round out the genetics and sort of
Hmm.
well, I guess maybe we'll transition to some of your field experiences after, but you've worked on ornate box turtles, Blanding's turtles,
Uh-huh.
population genetics,
Yeah.
and using those as sort of conservation tools. And I'm curious,
Hmm.
you've done some analyses looking at using effective population size to generate census population size estimates, and I'm the utility of that. That seems promising. Is it as good as a mark recapture study, though, in terms of
Mm.
the data you can get?
Yeah, I think I'm not quite sure how I want to answer that. I think you get different bits of information. Um, I've always been concerned with not with the concept of effective population size, but a little bit about its utility. It's in its traditional application, its utility in turtles, because I don't think with with few exceptions, most turtle populations, what you might describe as a reproducing population, are not going to be that big. And so you never really should expect that the effective population size that is problematic for turtles should be as large as what's applied to other species that have larger traditional demographic population sizes. Um, anyway, it's something that I've been pondering for the last decade or so. Something I started off thinking, and kind of being into the importance of effective population size and how problematic that can be with inbreeding and giving us a sense of that in turtles and populations. But the question is, to what extent does that effective population size being small tell us about the demographic challenges that that population is going to face? Is it going to really be, is the inbreeding going to have fitness consequences? Sorry, I'm not being very eloquent at this moment. um, and so to me, I'm at least as, I'm increasingly concerned that we can't just use the genetic markers as a proxy, I guess is what I'm going with this. And that what matters probably more is that we have to do capture mark recapture work to get at actual population structure.
Right, I guess that I've worked a bit with CJS models and
Okay.
some of those, and
Mhm.
just yeah, estimating like a parent survivorship, and I guess recruitment is the sort of
Right.
inverse to that in some ways. Like the how do you get that? I was struggling with that a little bit. It's just like how do you
Mhm.
get that from a genetic sample?
Oh, I see. How do you infer the demography from the
Yeah,
genetics?
yeah. I think that's
Okay. Yeah. Well, that's going to be math.
yeah,
Do we
yeah,
really want to go to math? I don't know if we want to do that. Um, but it's based on the principles of inheritance and allelic frequencies and all those other kind of things that kind of go into that.
right. Okay, so that but that's something. I mean, you're just saying that. I guess what I'm trying to get at is what
Oh,
is your
okay.
reservation? What sort of underlies the reservation is using the effective population size to infer the
Mhm.
the overall structure of the population.
I think to me it's because, or well, I don't know if it's about about the structure, but I'm thinking about it more because you were kind of alluding to the conservation side of things.
Okay.
And so for me, using the effective population size as a proxy for how concerned we should be with the population's stability, that's the part I'm less certain of than I used to be. I
Okay.
think with turtles, it's not such a tight relationship. And I don't know if that's because they regularly are small and therefore they're frequently purge deleterious alleles from their genomes. And therefore, when they have small population sizes, they aren't suffering fitness consequences like lots of other populations might when they go to a bottleneck of other species.
Okay. Okay. Yeah, that does make sense. So one of your analyses, there was a hazard function derived. I thought this
Mm,
was kind of interesting. Is it looking at the, I guess the just the mortality
yeah,
rate, sort of.
social hazards. Yeah.
Yeah, I'm
Hmm.
curious. I mean, it seemed like at one of the sites, the male that hazard
Hmm.
analysis, the slope was a lot higher on the line, but versus
Hmm.
another study wasn't the case. What was driving that, and what's the utility? The math is interesting. I mean, what's the utility of a
Hmm.
hazard function?
Yeah, the hazard function tells you, um, oh boy, yeah, it's not okay. So I have to say this is not my primary area of expertise. So
No
let me
worries.
start by
Yeah.
saying that. Um, um, that's why I work with lots of people. Most of them are all smarter than I am on these things. You have collaborators. But the hazard or that sort of concept, right, is about giving you some insight into as time goes by, essentially, how what's the probability of, well, I guess it's inverse probability of survival. And so when you look at things like some of the work we've done with painted turtles or snapping turtles or other species, um, from those capture marker capture data, you get a feel for, in our case, we were interested in sex specific sort of odds. Um, Anyways, it's just sort of a way of kind of giving us insight
Right. Right.
into that likelihood of survival. And having sex specific ones is kind of could potentially relate back to what we were talking about earlier about temperature dependent sex determination and the need to produce more or less of one sex in order to maintain a sort of a 1-to-1 sex ratio,
Right. Right.
right? So if males have a higher slope of their hazard relationship, that means there's a lot more turnover in that male side of the population.
Right, that makes sense. So that's yeah, something contributing to mortality, I guess you would
Hmm.
assume. Yeah, that's yeah, it's something I with especially in the past decade or so. It's the types of analyses you can do in terms of the mathematics behind it, all of that. It would be
Hmm.
impossible to be an expert in all of these things. It's a
Yeah, yeah. There's so many smart people in science now. Not that there haven't always been smart people, but there seems like there's so many more. Hard to keep up with the advances, whether they're quantitative or these other kinds of you know physical technologies. Mind-boggling.
Right. I mean, it's great. I think that you see there's more of this sort of melding of fields.
Mm,
You can
yeah.
pull in, especially like I guess like computer science, that kind of thing.
Hmm.
It's like these larger
Hmm.
scale uh, data analytics fields. You can pull that sort of thing in to get at these questions in ways that I mean you would know this more than I can is in terms because you've seen that evolution, right? But it just I mean,
Yeah.
yeah, what has it been like
Yeah.
to to integrate,
Yeah.
I guess, to adapt, right, to
Hmm.
these new technologies?
Yeah, I think it's great because I've always been, and for whatever reason, intrinsically about uh, being integrative, and I try to train my students the same way. Someone might want to come and they might want to just do animal behavior and so on and so forth. And I'm like, yeah, but you really want to have, this other skill set too, and whatever that might be. Almost invariably, I want them to come out with some quantitative skill. Uh, you know, really, really good at stats, or maybe they can program, or maybe they can do some theory, or maybe something along these lines, so that they're come out with a skill like that. And then also a combination usually between something that's field work, but also maybe bench. So maybe it's endocrinology, maybe it's something genomics, maybe it's just whatever it is. So so that's just individuals within my own team. And then so for me, like, there's only so many things that I sort of have a handle on. So yeah, I try to bring in collaborators, and they might be, again, people in my own lab who are just way smarter than I am about something, and that's a skill set we can leverage. Um, so I've seen that. I think one of the things that about the field in terms of technologies that's I don't know if it's underappreciated. Maybe it's more appreciated than ever, actually, in the last few years, since the unfortunate pandemic circumstances shut down a lot of active science, is the development of databases, and the I guess not so much, well, the development of them, but also the exploitation of them for answering questions. To me, that's been a really kind of cool addition to the field too.
Right, there's something recently like, Caleb's The Rosie
Yeah,
database.
yeah.
That is awesome.
Hmm.
It's just such a simple I mean, the concept is great. You can just keep adding data to but then to see, just to
Hmm.
see I guess, phylogram of all of the
Right? Isn't
evolution of
that
that,
cool?
it's
I love
fascinating.
that. Yeah,
Yeah,
yeah.
it's
Yeah,
awesome.
that was one of those. That's right, a classic, to me, a classic example of you know pandemic induced productivity to really make a useful sort of resource for the community of scientists.
Yeah, I mean, it's like and broad like broader level umbrella thinking. I think is like
Hmm.
this is useful to so many different people. It's
For sure.
it's yeah, it's great. Well, so I mean, there's always we've talked a lot about the things that you've done, but there's the side of things that are you don't capture that in a publication, and it's good to talk
Hmm.
about the research, the data. I think, but I'm curious about the more the side of things that goes into a lot of this, your field experiences. I guess, what I'm curious because you've done this for decades. Like, what day in the field stands out to you the most in terms of turtle analysis?
Huh. Hmm. Wow, that is such an interesting question. Uh, I I might have to. I think it's got to be the day that I kind of discovered what became Turtle Camp.
Okay.
So, so I'm. Can I tell you the story on that?
Yes. Yeah.
Okay. So, um, so I actually, when I went to, so I did my master's at Colorado State, and when I went to Chicago, University of Chicago, for my PhD work, I was planning to work on garter snakes and life history, sort of evolution in garter snakes. Um, this is relevant, trust me. But the point is, I needed some equipment to do the kind of work that I did, and we didn't get the grant, and I had to go through my qualifying exams very soon, and I had to come up with an alternative project. And so I thought, well, maybe I'll do something with these turtles. Um, but I need to figure out where I can collect eggs to do these kinds of experiments. And so I joined the Chicago Herpetological Society and started asking people there, what they knew about field sites for doing turtle work and all this kind of thing, and all that kind of stuff, and socializing and meeting a lot of really great, great herpers there. In fact, I still belong to the CHS. It's
That's
a great
awesome.
society. Um, but anyways, one of the persons kind of pointed me toward this site that was on the Mississippi River in Western Illinois, and so I had a buddy of mine, Gary Poxtus, who I mentioned earlier. At that time, he'd happened to move to Northern Illinois too. So, anyways, I picked him up. It was like a Friday afternoon, and we drove out there, and uh, we were looking at the sand prairie site because I was going to be wanting to collect snapping turtle eggs, and it looked really, really good. And I thought, wow, this is beautiful. It kind of reminds me of the sand hills of Nebraska, where I'd done my master's work. But the best part was we needed a place to spend the night, and where are we going to drop tent? And so we asked people in this little town, and they said, "Oh, just go down this street and hang a left, and there's this, Thompson Causeway and the Army Corps. They'll let you camp out on this island." And so we get out there and we go to drop tents, and it's like, I don't know, 5, 6 p.m. by this point, you know, and there are painted turtles nesting everywhere. It was like being dropped into a dream. It was unbelievable. There must have been 20 or 30 of them nesting. It was it was all over, and we Gary and I, we just looked at each other and we're like, "Dude, we're in turtle heaven." So that was really amazing. So yeah, that was long ago, but that was
That
great.
that's awesome. I mean, and
Yeah,
that ended up becoming, I mean, that a
yeah,
significant, yeah, right, right,
yeah, yeah. I wasn't seeking to start a long-term project. It kind of, revealed itself, I guess.
right, right. That is awesome. That is really cool. In turtle camp, I mean, this has been something we
Mhm.
haven't talked about this yet, but
Yeah.
it's really cool what you've been able to do with this and like sharing the
Mm.
how fascinating turtles are. What stands out to you from that?
Yeah, it's evolved as many of these things do through time, and I think the thing though that it was sort of a macroevolutionary jump was when we started. And I attribute this to several of my students at the time: Laurie Newmanly, Shannon Thal, and Jeremy Strickland, who really made it happen. Where we started to get the high school students come out, and it became, and they would come for like a 2 week period, and we were recruiting students from all over, people from rural environments, urban, whatever it was, and making contacts to try to find students that might be interested and good at biology, but had, no outlet for that. And so we talked to teachers, and sometimes in cities like in Chicago, Jeremy would talk to their local pastors, who kind of knew who were the good kids around in the neighborhoods and stuff. And so we just recruited kids from all over that way. Um, Again, rural and urban type environments. Anyway, so it was fun to have these high school kids come out, and we started, and it was relatively easy to get money to support them. And for them, this was, well, I guess ultimately for me too, transformative as an experience. Like a lot of them hadn't seen any of this kind of biodiversity before. They'd never been in a tent cooking over a fire. Are you kidding me? And, watching turtles nest and seeing, you know, because I've seen it thousands of times, turtles nesting, right? But seeing it through their eyes, mind blowing. And it's really, anyway, for me, a transformative thing. We did that for a good decade or so before I ended up moving here to Michigan State. So that was really, really great. And yeah, I don't know how many of those students, but I think a lot of them went on to community college or university. Um, I know at least one of them is pursuing a PhD right now at UMass studying sea turtles.
That's
Um,
awesome.
so, yeah, pretty fun. So I would say that.
That's awesome. Just the holistic. I mean, it's so
Hmm.
fun. Is I think people that just like genuinely, it's just fascinated by the turtles. It's so fun to share
Mm,
that interest with
yeah.
people, especially young people. Is I've gotten to
Yeah.
the Turtle Conservancy. I grew up in
Uh-huh.
Southern California, so I
Oh,
would go
yeah,
up
yeah.
there a lot, and I spent a lot of time there on different projects with them, and a lot of times they would have the local schools come out with the kids, and just to see
Yeah.
a lot of these places are sort of underserved, and just to
Mhm,
see
mhm.
all these people just engaged with nature and so interested by it is
Yeah.
it's just really
Mhm,
inspiring, even as another young person, just to
mhm.
see other young people get
Right?
really interested
Yeah.
is
I
it's
love
great.
that. Yeah,
Yeah,
it's awesome. And we even see it too because now there's a lot of people from the public that camp at the turtle camp site. So a lot of retired folks with their grandkids. So we're getting these multi-generational, hey, what are you doing? Kind of things
yeah,
while we're watching a turtle nest, and they're really fascinated by the science, but also by the natural history of, wait, she is leaving? She's not going to take care of her babies, all that kind of fun stuff. So
right,
yeah,
right.
get a lot of great questions.
That's great. Well, so I guess just sort of the last thing, and then we can sort of
Mhm,
wrap things up. But I'm curious, holistically, and all the adventures that you've had,
mhm.
what's the story, the adventure story? I was actually at a conference with Kurt Buellman last week,
Oh, yeah.
and your name came up, and he was talking about this trip you both did in Australia. I so that's
93, 94, yeah,
that
that
sounded
was great.
that sounded interesting, but I guess just broadly, maybe that,
Mhm.
but whatever stands out to you as your most
Oh,
interesting
man.
adventure.
Yeah, not necessarily a turtle adventure, a
It can be
herpetological
whatever.
adventure.
I mean,
Yeah. Oh, man. Yeah. Well, let's see. Yeah, I don't know if I can choose a most interesting one, but I can choose one that's at least, I think, interesting. How about that? Um, I'll tell you that. So this is actually one, this involves snakes, so it's a little bit off topic. Um, but I was so Butch Brody and I were in Costa Rica, actually at La Selva Biological Station. We were co-instructing an organization for tropical studies course for undergraduate students, or no, sorry, for graduate students. And but so we were doing a project on coral snake mimicry at the time, and I had done with using these plasticine snake models, and I had done some with these plasticine turtle models to look at differential predation on plasticine turtles of different sizes. So there was a turtle connection here. But at the time, we were doing this snake project, and the students had their own individual research project that they were doing for the course itself, while we were doing this other side coral snake stuff. Um, and at the end of the day, this is, 19, gosh, it might have been 92. Um, so the students, in order to analyze their data and for us to help them, had to come to this one spot at the station. And when they would do it in the evening after the end of the data collecting during the day. And the point being, you know, I was helping with the data, and the students would come in and they would tell us if they've seen, snakes, so that we could find them for our coral snake mimicry project. And the student comes rushing in and says, Dr. Jansen, Dr. Jansen, I see the snake, it's down next to the path. You want to come and check it out? So yeah, I race out, and I'm wearing shorts and flip flops, and I don't even know if I had a shirt on, but I raced out, and sure enough, there it was in the leaf litter next to the trail. And I look at it, and I can see that it's black and white banded. So I knew it wasn't a coral snake, but it was some kind of mimic. But I hadn't seen it before, and I thought, I got to catch this because Butch is in here right now. He went back to get the models for us to place in. So, I mean, so I step off the path, try to catch it. It strikes at me and misses, and it slithers off. And I'm like, unbelievable. How did I miss that? And this is a snake that's like a good meter long. So it's not small. So how did I miss it? I don't know. So I'm like, okay, I can't let this get away. So I start walking off the path a little farther, a few more steps, and I see it at the base of what's called a walking palm that kind of has these roots that are up above, and it's kind of woven in around a couple of them, and it's starting to go down into a hole underneath. So I quick grab it from like mid body and start, threading it back through until I got it all the way out. And so now I've got this thing, black and white band snake, in my hands. I walk back to the path, um, and I have no idea what this thing is. So I put it into a bag and take it back to the station. I figure we'll look at it, we'll go through the dichotomous key, there's no internet at this time, by the way, either. It didn't even exist in '92, probably. And so what are we using this dichotomous key? And we go through it, and I'm holding the snake, and the students are telling me what the number of scale rows at mid body, and so on and so forth. Anyways, we keep coming back to presence and absence of a L'Oreal scale, which is the key character for determining whether or not it's a coral snake.
Yeah.
So, we do this like 3 or 4 times. I keep saying it's a coral snake. I'm like, "It can't be. It's too big. It's black and white banded. The only coral snakes here are either red and black or red, black, and yellow, right? Tricolors. So I don't know what this is. We'll put this in a Ziploc, and I'll wait for Butch to show up." Butch shows up about 10 minutes later, and I say, "Dude, I caught this snake, but I have no idea what this thing is. It keeps saying it's a coral snake, but I don't think so because it's black and white banded." He picks up the bag, he looks at it, he looks at me, he goes, "Dude, man, that is a coral snake." So I've been free handling this coral snake. It turns out it's a morph that had never been described before.
Oh
And there
man.
was no antivenin. There is no antivenin. So that's my story. I should have been dead, I guess.
And that's some serious. I mean, you've got
Stupidity.
yeah, that that you've got what, a few hours or so. I mean that's a
Yeah.
serious generally.
Yeah.
That's
Yeah.
crazy. Yeah.
Yeah. Just when you think you're an expert. Oof.
Well, yeah, that's my logic. I've spent a decent amount of time in Belize. I'm going back for
Ah,
my 4th summer,
nice.
uh, at some point here, but
Mhm.
there I don't mess with any. I won't even touch if I know what a
Yeah.
snake is, I won't even touch it because I don't know. I just try to avoid.
Yeah.
I yeah, I it just
Not take a chance.
yeah, I'm not taking a
Yeah.
chance. I had kind of a similar situation this summer.
Mm.
Uh, there was someone actually with our group that was holding a young coral snake in their hand, like palming it, and again, the same thing that the person they were with
Mm.
said it's not, and then they came back to the camp and
Mm.
showed a picture without it in the hand,
Yeah.
and the guy's like, "Oh, that's a cool coral snake," and then he flips to the photos of him holding it. It's like, but the one
Mm.
this was crazy. In Belize, we
Mhm.
work on this river, the Bladen River. It's a crystal
Mm.
clear river. So we went
Mm.
snorkeling at night to look for
Ah.
the Sturritipus,
Yeah,
the
yeah.
Mexican giant musk. And so we're snorkeling there at night, maybe 30 minutes, pitch black. I've just got a light, nothing else. And
Mhm.
I slam into something on the top of the water. I hit something really long.
Mm.
And I'm thinking, okay, this is what I just hit like a nutria or some kind of mammal.
Mhm. Yeah.
And I shoot down on the water and jump back up after a
Mhm.
second just to make sure. And I swim over the side, and there's a 5-foot long. It was the biggest fer-de-lance I've ever seen, just sitting on the side of a rock wall, and
Oh,
it had been sitting out
wow.
in the water. I mean, I guess that they this is like
Weird.
the only place they do this in Belize.
Huh.
It's a
Yeah, I've
rare
never
thing to
heard of that.
see.
Okay.
Yeah,
Huh.
but so that was there's nothing like I can only imagine what you felt
Yeah.
after knowing it
Ah,
was a coral snake.
yeah.
You just knowing you touched something like that
Yeah.
is yeah.
Right. And that it had struck at me, and, we were there. I never would have I probably wouldn't even have lived till we got to San Jose if it had envenomated me. Yeah, would have been bad.
That's
Yeah.
that is
My
wild.
life flashed before my eyes for sure.
That is cra yeah, it's a shocking experience. Well, that I guess maybe that's some kind of initiation. You have to have a run-in with a venomous
Okay. I guess.
I
Yeah.
don't
Not
know.
by choice. Certainly not by
Yeah,
choice. I never would have knowingly done that.
right. Well, that's a really good we've had a decent number of snake stories, so that's a good that's a good one, though. That's good.
Mm,
So I guess just to close this off, I mean,
mhm,
the thing that, as students, a lot of students listen to this, have reached out, that kind of thing.
yeah.
I'm genuinely curious about, what is one piece of advice you would give for
Mm.
someone looking to make turtle work a career?
Yeah, you've got to have perseverance and passion. So you've got to be able to work hard. You cannot give up. Um, and it's interesting to me. I've had, as you pointed out at the beginning, 30 some graduate students. I don't know if any of my graduate students still work on turtles. It's hard. It is hard to make it a career. And not because, yeah, anyways. and we need more turtle biologists. So it's just not like it's somehow not necessary. It is increasingly crucial that we have folks who are passionate um, about doing something with turtles. It doesn't have to just be conservation. Just listen to me. It doesn't have to be conservation alone, but working with them. Um, so I really think that being persistent, not giving up, and also maintaining that passion, because like anything, there are going to be times when it's rough. That's the way life is. Times when things are awesome, but you've got to push through when it's not great.
Awesome. Well, I think that's just good general life advice. So
It
it's
is, yeah. You don't have
yeah,
to be a genius. You don't have to have any of this other kind of things. You just have to want it.
right. Well, that's all. I mean, I'll take it to heart, and I hope people listening will do the same. That's awesome.
Yeah.
And I guess yeah, the last thing that we like to finish off with. Normally, we've got
Mm.
not normally, but a lot of times with a lot of people. But
Mhm.
we just a quick round of turtle trivia. So I don't know if you've got
Oh
a
man.
random turtle fact. Make it as obscure as possible, or
Oh
whatever,
man.
and you want to try to stump me with it. We could do
Mm.
that.
Oh man. Gee whiz. I didn't prepare for that. I have to think. Um, oh
It's always
man.
yeah,
I
yeah,
don't I'm so sorry. I can't think of anything that I think would be really interesting and stumpable.
no, it's yeah,
Hmm.
it's all good. I mean, I could throw something your way if you wanted.
Sure. Sure.
All right. I guess an evolutionary.
Mm,
Trying to think of a good evolutionary question. I've got two
mm.
questions, two random
Okay.
questions. So okay, with the map turtles, the graptomes,
Mm,
that they're a great example of sexual size dimorphism,
mhm,
and there's been some work done. This is sort of in your vein of it, but it's outside of what you've done. So it's like I don't know if you,
mhm.
but there's been some work done to look at how that magnitude of sexual size dimorphism has evolved.
Yeah.
I guess there are two possible. It's a
Mhm.
50% chance. How is it? Have the males gotten smaller,
Mm,
or have the females gotten larger? How is that?
mm.
What has happened with the magnitude?
Yeah, I think the work I think it's Jeff Lovich did it, I think. My recollection is that the females are gigantized? I don't know if that's a word. Um, because a lot of them, many of them are sort of clam crushers, so to speak. And so they have these megalocephalic, not every species does, but many of them have megalocephalic females. Well, that's what I think. But I
Yeah,
don't know if I'm right.
it's possible there's something. I mean, with a question like that, it's possible he has. I'm thinking of Peter Lindemann, his
Oh,
work.
Peter. Yeah, Peter
Yeah,
too. Right.
he did
Yeah,
some work with.
that's right.
I think it was looking at independent contrasts on the phylogeny, and
Oh,
he figured out using those
uh-huh.
how the magnitude of change in the size has shifted. I think his conclusion was that actually the males have
Oh,
gotten smaller.
miniaturized.
So the males have gotten
Really?
smaller, but it's possible that's changed, but that was what his
Ah,
analysis said.
you
Uh,
stumped me then.
yeah,
I
I
wouldn't
mean,
have guessed it.
it's an obscure
That's a good
thing,
one. Yeah.
but, I mean, you had like, the saying that they're larger, I mean, that's right, so yeah, it's and then I guess the second one, this is just a random this is
Mm,
a big analysis I just, we just submitted this actually to a big journal to look at
mm,
just the total turtle diversity. But how many turtles does
mm.
okay, this is better. The state of Alabama has the most turtles in the United
Ah.
States, but how does that compare to countries, global countries?
Oh, yeah. The Southeast is such a hot spot for herps, salamanders too. Um,
Including sea turtles too. So all
well,
turtles.
right. Yeah, Alabama's got a coast that helps. Oof. So if Alabama, basically you're saying if Alabama were a country, where would it rank?
Yes, exactly. Yeah.
Yeah, I think about that. I'm thinking, wow, It's got to be way up there. Because if you think about, yeah, I bet it's top 10. I bet there aren't too many. If you're thinking like Central and South America might be the ones that might be above it, but I don't think there's any others. I don't even think Australia. Well, oh, what am I thinking about? Asia. God, what an idiot. Right. Yeah. There are some East Asian ones that might be high in there. Anyway, I'm going to say top 10.
Yeah, that's right. It's fourth based on this analysis.
Whoa.
Yes, yeah.
Wow. I would have thought it would have been closer to 10. That's impressive.
Yeah, yeah. So yeah, it really was. I did not expect
No.
when we put all these metrics in to see that pan out. But yeah,
Wow.
you've got well, North America, the United States has more, but that sort of is like,
Yeah.
yeah, obviously. Uh,
Yeah.
Indonesia,
Yeah.
Mexico, and oh man, what's the Indonesia, Mexico, and there's one other. I can't
Huh.
recall off the top of my head. But yeah, it's top four. So
Wow.
it really, yeah,
Interesting.
it's pretty incredible. But
Yeah. Definitely would have thought. I mean, it's interesting that Mexico's there. I guess it covers a large space. I would have thought some of the Central American countries and then some of those East Asian ones for sure. Yeah.
yeah, it's very
Huh.
close. So it's like this tally was fun to put together because it's like
Hmm.
Alabama's 39 tax, Mexico's like 60 something, and then Indonesia's
Wow.
like
Yeah.
41,
Yeah.
and then under all a lot of the Central
Yeah.
American, Asian countries are between 36 and 33. So
Okay.
it's
So there's
it's
a bit
very
of a plateau there. Yeah.
yes,
Okay.
yeah. So,
Cool. That's
but
so
yeah,
cool.
yeah, it's just a kind of a random trivia fact, but yeah.
Yeah. Oh, I love that. Well, hopefully that'll yeah, hopefully that'll get published because that's a fascinating result.
Yeah, it's this thing that we talked to Russ Mittermeier's the
Oh
turtle
yeah.
conservation
Hmm.
guy on the podcast, and he had a lot of ideas and kind of made this thing
Mm,
happen. And uh,
good.
so yeah, we put all that together, and it's a cool mix of. I think anyone can understand it, but it also kind
Yeah.
of demonstrates how incredible the Southeast is. So it's good.
Yeah, and that's good. It has other benefits of generating local pride and things along those lines, and maybe people pay more attention to taking care of what they've got when they know it's such a globally valuable resource.
Yeah, yeah, exactly, and yeah, just using it as a way to foster it with the federal issues and everything. It's federal funding can
Yeah.
go away in a second, but
Right.
it's like using this as a way to foster private
Hmm.
initiative, private investment in
Hmm.
turtle conservation. Like, here's this incredible diversity. We need to
Right.
take pride in this and support it
Absolutely.
financially in any way possible. So yeah.
Absolutely. Yeah, for sure. No, that's great.
Yeah. Well, that's awesome. I mean, this has been an incredible conversation.
Yeah,
Thank
a
you
lot of fun.
again. And
Mhm.
I just say the floor is yours if there's any last minute things.
No, I mean, with you mentioned, you alluded to, the loss of federal funding, and I think there's an increasing skepticism, and maybe it's global, for sure it's in this country, about science and the value of what we do. Um, I guess I would leave it with, I don't want folks to give up hope. I want people to have hope because I do think no matter what happens, science is always going to exist. Science is a way of knowing the world, right? It's a process. We're always iterating. We're always doing better. Sometimes, we think we have an answer, but yeah, and we need more people to help us continue to figure out, how the world works. So that's my uplifting thing. I know a lot of people are trying to bail on science. Don't do it. If you have a passion for science, please stick with it. There will be a place for you.
That's awesome. Well, that's a great place to end. Thank you, Dr. Jansen. This has been awesome.
Yeah,
And we will see
likewise.
everyone on the next one.